Olga Lazareva, Technical University of Munich. My talk title is BiCoN, Network Constrained by Clustering of Patients and Omics Data. Unsupervised learning approaches are crucial for biomarkers discovery, as they are entirely data-driven and not biased by our current understanding of diseases. But classical clustering methods do not allow to cluster genes and patients simultaneously, while also providing mechanistic explanation of the results. To bridge this gap, we developed BiCoN that takes as an input a gene expression matrix and an interaction network and selects genes which are the most valuable for patient clustering, while also connected in the PPI network. BiCoN constructs a bipartite graph from gene expression matrix and then adds edges between genes based on a PPI network. Then we use a combination of ant colony optimization and local search algorithms for robust and fast subnetwork extraction and patient clustering. Our objective function is constructed such that the extracted subnetworks have the highest possible difference in gene expression. We applied BiCoN to the TCGA breast cancer dataset and expected BiCoN to be able to recover known subtypes assigned with the gold standard PAM50 gene panel. For the analysis, we focused on patients with the most common molecular subtypes, luminal A, luminal B, and basal. We first show on panel 1 that BiCoN can separate patients into the two clinically well-distinguishable subtypes, luminal and basal. As expected, the separation is straightforward and the clusters correspond to the subtype labels. Then we considered only patients that were originally classified as luminal to see if we can further stratify them into subtypes luminal A and luminal B, which are known to be difficult to separate on the level of gene expression. Our solution on panel 2 did not really agree with the PAM50 classes, but the derived subnetworks show robust correlation with immune system response function, which is reasonable given that tumor samples are infiltrated with leukocytes. This demonstrates that BiCoN can find an interpretable difference between groups of patients, even without being explicitly asked for it. The next question is if BiCoN is better in this task than other unsupervised methods. On panel 3, we can see that BiCoN performs as well as other methods like hierarchical clustering, but also finds subnetworks which explain subtype differences with a vastly lower number of genes than classical clustering methods. Network incorporation not only makes BiCoN more interpretable, but also leads to better robustness and, in particular, batch effect robustness. To demonstrate this, we simulated two datasets such that the signal for a batch effect is twice as strong as the true biological signal. The genes that carry that true signal were connected with one another in a simulated Barabasi-Albert graph. We have simulated 10 datasets with batch effect, as I described, and evaluated the performance of 3 popular biclustering methods and 2 clustering methods. In the end, only one biclustering method has produced any results. Also, to make sure that we do not favor BiCoN by enforcing connectivity of the foreground genes, we performed a simulation with a Barabasi-Albert graph where foreground genes were randomly distributed and not necessarily connected. In both scenarios, BiCoN performs better than other methods, as you can see in panel 4. In conclusion, BiCoN can cluster patients and extract subnetworks that can mechanistically explain the patient stratification. Moreover, simultaneous clustering of gene expression and networks makes BiCoN robust to noise and more robust to batch effects than typical clustering and biclustering methods. You can follow the QR code to get to our webpage, where you can find our preprint, the web interface, and the PyPy package. Thank you for your attention. Do you have any questions?